Hello. Today's lecture is going to cover gene editing technology. What is it and how can it help? By Engdao Fan. As we have learned, modifying genes of living organisms is nothing new. However, new methods for gene editing technology has significantly improved. What is gene editing exactly? Simply put, it is the modification of genes in living organisms. This area of research allows scientists to continue to develop methodology to 1, further understand gene function, and 2, to find ways to use gene editing as a therapeutic to treat genetic or acquired diseases such as leukemia and HIV. Gene editing technology can be used to correct, introduce, or delete targeted DNA sequences. Modern-day gene editing technology includes the first one being zinc finger nucleases, which was discovered in 1985. ZFNs represent a revolutionary change in gene editing technology. However, they are hard to design for efficient cutting at the desired site. They take months to design and can still lead to insufficient results. Next, we have transcription activator-like effector nucleases, or TALEN, which were discovered in 2011, but their size is much larger than ZFNs, proving more difficult to deliver and express. Third, we have the clustered regularly interspaced short palindromic repeats, or CRISPR. It has been an ongoing process in its discovery. Principles of how they work was first discovered in 1993. In 2012, how CRISPR's work was solidified. This system can precisely cut DNA at a targeted area. The DNA segment that is cut is then saved as a memory to fight future infections. Of the three, CRISPR technology is the most effective and costs less to create. Why use gene editing technology? Well, scientists use this technology to gain further understanding of how genes function, which then lead to an understanding of underlying genetic causes. This understanding then leads to scientists figuring out ways to use gene editing technology as therapy and treatment of diseases. Outside of the clinical setting, scientists use gene editing technology in their continued research to make and improve biofuels in agricultural crops. We've all heard of the term GMO. That's the same thing. So how did we get here? Let's rewind to a century ago to when Gregor Mendel worked with crossing a tall pea plant with a short pea plant. Though this has nothing to do with gene editing as we know it, it is important to mention his contributions to science as he laid the foundation of what will become known as genetics. Fast forward to the 1950s, when James Watson and Francis Crick, with the help of Rosalind Franklin, discovered DNA's shape is a double helix. This discovery opens the floodgates for DNA research. In 1968, Werner Arbour discovers restriction enzymes, which will later be used in gene editing technology. Arbour observed that bacterial strains were able to fight off bacteriophage infections by cutting off its DNA. He tested his hypothesis using E. coli and found that these cells produced two enzymes, one being a restrictive enzyme, which could identify and cut foreign DNA, and the second being a modification enzyme, which recognizes the host DNA and protects it from cleavage or being severed. Since Arbour's discovery in 1968, the scientific community continues to thrive, especially in the field of genetics. With the discovery of ZFNs in 1985, scientists have been able to develop gene-targeting drugs, such as Glyvec. Glyvec is a cancer treatment drug that is still used today to treat myelogenous leukemia. In 2017, 2 CAR T cell therapies were approved to treat acute lymphoblastic leukemia in children and advanced lymphoma in adults. Using this treatment could possibly replace chemotherapy as the go-to cancer treatment, as CAR T cells are non-toxic to the human body. In 2020, scientist Dr. Javisha Bajaj and a team of researchers at the University of California, San Diego School of Medicine discover a new gene called Staufen2, which regulates and drives the molecular forces for leukemia stem cells. These cells are responsible for reproduction of the disease and its therapy resistance. Prior to this discovery, they had seen Staufen2 in the brain and spinal cord, but had no idea of its function. The continued research in leukemia is important because the survival rate is less than 10% in its most aggressive forms. In conclusion, gene editing is simply the modification of genes in living organisms. Gene editing technology is having the ability to introduce protein nucleases into cells that target a specific DNA sequence, cut it, and allow the cell to repair itself without the mutation. From animal breeders and farmers modifying their crops and animals to create the perfect crop or breed, to finding ways to combat acute or chronic diseases, gene editing technology continues to make its place in the world.